With that, I'm excited to introduce our speaker. And I will also just remind you, as you probably know if you're here, that this is a weekly event. And I believe we have another talk coming up next week and the following week, so Tuesdays at noon. But today we are really excited to welcome distinguished Professor Emeritus J. Clark Lagarius. And Professor Lagarius, please let me know before I do the rest of the introduction if I am butchering your last name. Oh, you're muted. Sorry, I can see that you're speaking, but I think you're muted, so I can't hear you.
Um, I usually use Lagarius, but
Lagarius, thank
Lagarius
you.
is a Greek pronunciation, so it's okay too.
I would like to use what you usually use, Lagarius. So please
Yeah,
forgive me.
no problem.
Professor Lagarius is in the Department of Molecular and Cellular Biology at UC Davis. He earned his bachelor's degree in botany and chemistry in 1975 and his doctorate in chemistry in 1979, all from the University of California at Berkeley. A faculty member at the University of California at Davis since 1980, Lagarius is known for his work to understand the molecular basis of plant, algae, and cyanobacteria light perception, focusing on a group of linear tetrapyrrole-based light sensors. Again, forgive
That's good.
poor pronunciation. Uh, in the phytochrome superfamily, the author of 6 issued U.S. patents and over 175 peer-reviewed publications, Lagarius and colleagues' research has provided new knowledge impacting crop improvement efforts, synthetic biology applications, and development of bilin-based imaging and optogenetic agents for biomedical and agricultural applications used by many researchers worldwide. Thank you for joining us today. We're really looking forward to the talk. And please feel free to make any amendments to the bio that I shared.
No, that's fine.
Okay.
Thank you so much. And it's an honor to speak with you. Part of the agricultural experiment station appointment that I had for many years is outreach to applications in agriculture. And so I'm honored to be speaking to you and, provide you with a glimpse of some of the stuff that I've been interested in for 40 years, actually for even more. When I grew up in Western Pennsylvania, I was out there looking at plants coming up, crocuses coming up early in the spring through the snow. And I've always been really interested in how plants develop. And so I'm interested in why plants need to see. So we all see. Why do plants need to see? Um, and I've had help with some of the slides with Julian Malouf, who's a faculty member in plant biology here at Davis. So I've pirated some of his slides. We've both given talks at various meetings. And I'm sharing some of his, so I want to acknowledge that. Let's see if I can get this to move. I don't know why it's not moving. Okay, so for plants, light is food. And so this particular cartoon really illustrates it nicely. When a friend is going, "What you doing, Mooch?" I'm trying my hand at synthesis. And how's it going? And he says, "You're blocking my lunch." And so light is food. Obviously, um, animals can walk to forage, but plants can't. Plants have to grow to better foraging if the light conditions are not optimal for them. And so the only way they can do is to either grow toward the light or away from too much light, or flower, make a seed, and have that disperse to a better light environment. So As a consequence, plant growth and development is extensively regulated by the environment, particularly by the light environment, but also by temperature and other environmental biotic and abiotic events. So that's really what I'm going to be talking about, is that the growth and development of plants is plastic. Unlike people, plants, where they ended up rooting themselves in the ground, they're at the mercy of that environment, and they're going to grow differently depending on where they land. Okay, now. Okay, so plants monitor many aspects of their light environment. As shown here is mung beans grown either in full darkness on the left here, or grown in the light. And you can see that they're quite different. And these seedlings are monitor not only the intensity of light, but the color, known as the quality. So what the spectral distribution of the light. They can monitor direction, and they can monitor the timing of the light, so the length of the day and the length of the night. And so all of these aspects contribute to the development. You can see in this drastic conditions that seed plants require light for the synthesis of chlorophyll. So you can see this plant that's grown in full sunlight is dark green, whereas this dark grown plant does not make chlorophyll, and it's growing much taller, and it's protecting its initial leaves as it tries to grow through soil looking for light. Of course, light is required for photosynthesis, and I'm not going to be talking about light that's harvested for photosynthesis, which fixes carbon and contributes to the biomass. But light also impacts photosynthesis because of the way the plant architecture receives and adapts to the light that it gets. So we in the field of photobiology, as I'm known, perform experiments called action spectroscopy. And basically what action spectroscopy allows you to do is take a large, this is the Okazaki spectrograph, which is a huge grating with a giant incandescent lamp that is refracted into individual things, and they put plants under different colors of light that have been diffracted, and they measure particular responses: growth, chlorophyll synthesis, photosynthesis, and development under different colors: red, green, blue, the whole spectrum of visible lights. And they measure how much light is required at each different color. And from that, they, as you can see here, here's the visible light spectrum. They can find the sensitivity for a particular response for each color of light. And they use this information to determine whether or not this light is, whether blue light is responsible, or green light, or red light, or some combination of those. And that gives you hints of what is perceiving the light, the color of light. And the color of light is what I'm going to really talk to you about mainly today. So light regulates many aspects of development. So let's start with the very beginning of development, seed germination. And light regulates seed germination. So many of you, when you dig up the ground and you unearth seeds that are buried in the ground, you see a lot of weeds starting to germinate. And these seeds have received light. And actually, an action spectrum showed for particularly small seeds, which have very little seed reserves, such as lettuce or Arabidopsis, which isn't really a crop. But let's take lettuce here. Lettuce seeds, if you just try to imbibe and germinate them in the dark, very few of them will germinate. Some will germinate, but if you grow them under red light or give a pulse of red light and then put them in darkness, and if 4 or 5 days later measure seed germination, you can see that red light has promoted this seed germination. But if you do this under far red, which is longer wavelength red, so close to the near infrared, the seeds don't germinate. And you can do this experiment where you just give red followed by far red and then give another red, the seeds germinate. But if you add another far red, the seeds don't germinate. And this experiment was one of the classic experiments that showed that there's a photo switchable pigment, a pigment that absorbs red light and far red light. Each wavelength of light could switch it from on or to off. And that's the phytochrome system, which is what I'm going to study. And I'll give you a little bit more information as I go on in my talk. So this type of experiment, seed germination, is also promoted by other wavelengths of light, but I'm just going to talk about red and far red light for this purpose here. So seedling emergence, light is a cue for that. And so as I showed earlier in that mung bean slide, the mung bean that's grown underneath the soil, if it has germinated under there, and it's going to grow elongated through the soil with a hook that protects the cotyledons and the primary leaves from damage as it forages for light through the soil. And this, in dark-grown seedlings, this is known as etiolated growth, where the apical hook is shown, and this is for dicot plants, not monocot plants. And the cotyledons are unexpanded, and the hypocotyl, in this case, is very elongated and elongates so that the seeds or the primary leaves, when they reach the surface, can perceive the light and also make chlorophyll, because chlorophyll, remember, requires light to make it, and you need chlorophyll for photosynthesis. So light causes what we know as de-etiolation. So as the plant etiolates through the soil, as it's getting closer and closer to the soil, it's getting more filtered light until it finally sort of emerges or is close to the surface where it gets sufficient light. This causes an inhibition of growth. So the hypocotyl growth ceases and the hook straightens out. So this bends around and the hook straightens and the cotyledons expand and green and the primary leaves start to produce and develop further for photosynthetic development. Until it receives light, it's growing on this seed reserve, which is mostly in the cotyledons. And so all the growth is used, all the growth requires fixed carbon that's already present in the seed. And until it gets light, so it's doing this last gasp of growth. If it doesn't get light at a certain point, then it can't photosynthesize and it runs out of seed reserve. It runs out of food, basically. And that really depends on the different seeds you have and how much seed reserves present and that sort of thing. So you can imagine a lettuce seed, if it's way down there, doesn't have much to sustain development through any really thick part of soil. So those seeds will essentially not make it unless the soil's turned over. Okay. So that's seedling emergence is the second facet of plant development that's light dependent. Now, the third, which many of you probably already know or familiar with, is phototropism. And phototropism, shown here with a dicot plant, a mustard seedling on the left. And now this is maize on the right, maize coleoptiles, bending toward this response. Phototropism occurs in all plants, and it is responsive to blue light. So red and far red, which I talked to you about in terms of seed germination, in this case, phototropism is a blue light response. So plants have eyes for blue, plants have eyes for red and far red, and so do people. We have eyes for, we have rhodopsins for red, we have rhodopsins for green, and rhodopsins for blue. So the pigments that plants use are different, and they're not in any specialized organelle. So there's no eyeballs in plants. Essentially, every cell in the plant is actually perceiving light. And of course, every cell that's on the surface can perceive light, but the roots are, of course, are down under the soil, and only when they get exposed, roots will even respond to light, often to just grow away from the light so that they stay submerged. Again, that's phototropism dependent. This is another response that is what plants see. And this response is something that affects intracellular orientation, arrangement of the chloroplasts. And the chloroplasts are the packets in the cell that are photosynthesizing and fixing carbon. And if you take a plant and put it in the dark for a while, the chloroplasts will just sort of evenly distribute throughout the cell. But now you give a pulse of light, and what you find, if it's low amount of blue light, LB for 60, you see that the chloroplasts, more will be accumulating in this clear spot of the cell. If you use high blue light, which can be damaging to the light, the chloroplasts move away from this and they try to hide underneath each other to prevent damage, sacrificing, if you will, the chloroplasts that's on the top and shading those below to prevent them from damaging. So again, this is designed to optimize light capture for photosynthesis. And again, this is a blue light response. So the amount of growth of a adult plant, you're probably all familiar with plants grown in the shade versus plants grown in full sunlight. They grow differently. The plant looks quite differently depending on the variety of the plant and the genetics, but wild type plants tend to want to grow taller to avoid and compete for light faster than the plants that are growing next to them. And so the stems and petioles of the leaves tend to elongate if they're in shade. And the resource allocation within the plant, so the amount of carbon that's sent to the leaves, roots, shoot, seed, and fruit, all this resource allocation changes depending on whether the plant is grown in the shade or whether the plant is grown in the light. So this affects the extent of organ development, fruits, seed yield, and that sort of thing. And the other thing that light sensing does is it affects whether or not this plant either grows, so it's foraging, or whether it's defending itself. And so it affects its ability to resist insectivory and also microbial pathogens. And so generally plants that are grown in the shade tend to be, they grow, and plants grown in full sun tend to be more in defensive mode and more resistant to insectivory and other pests. These together, this process is known as shade avoidance. And shade avoidance in the extreme sense is if the plant is in prolonged shade or it's a plant that germinates underneath a deep canopy cover of a forest, the only thing the plant can do to get out of there is to flower. And so flowering timing is clearly affected by shade versus light, full sunlight. And here's an example of shade foraging. This is genetically identical individuals of tomato. This is grown in full sun, and this is grown in the shade of simulated shade, in this case, of the light that would be transmitted through a canopy of other chlorophyll-containing organisms. And the most competition plants have is with other plants. So most of what they're sensing is the depletion of light that chlorophyll absorbs. So chlorophyll sucks out all that red and blue light, and the spectrum is quite different. And in shade it's greenish, so it's transmitting green, but the intensity of red and blue is severely compromised. So this is a real problem for high crop densities. And so if one plant wants to get more yield per unit acre, one would try to plant closer together. And unfortunately, shade avoidance is counter helpful to this process, because shade avoidance, the plants try to outgrow each other. And in a monoculture, this isn't really good, because the arms race for growth puts all this energy in the stem elongation, and in general, it doesn't fix biomass that helps support a robust seed yield or fruit yield in this case. So, of course, these tall and spindly plants tend to blow over in the wind, and so they have all sorts of problems. And most, if not all, agricultural genetic breeding programs have sought plants that typically dwarf in stature, which have lost this shade avoidance behavior through the process of affecting either the photoreceptors or the signaling downstream of the photoreceptor that allows plants to grow this way rather than responding to shade. And there are some natural plant varieties that are shade tolerant species that do not show shade and foraging conditions. And many of these are natural plant varieties that are adapted to living in the forest understory. Again, in both cases, photoreceptors are important for this. So what are the photoreceptors? Oh yeah, the last thing I want to mention is light regulates flowering time, as I was saying. So many plants use light to determine the day length, which obviously is changing during the seasons. So plants grown at the equator don't experience a lot of variation in the day length, but plants further from the equator at higher latitudes experience seasonal variation of the day. And actually what they do is they measure the night. So plants that are long day plants start flowering when the days are longer or the nights are actually shorter. And so you see these plants will tend to flower toward the summer period. But short day plants are plants that are induced to flower when the days get shorter. So the plants will grow robustly during the summer, and as the days get shorter by the fall, they're induced. They measure this long night period, and that leads to initiate flowering. And of course, there's all sorts of interest in understanding the photoreceptors, which are regulating this. And in fact, my lab is quite interested in looking at different kinds of alleles of the photoreceptors that affect this flowering photoperiod sensitivity. Photoperiod also regulates bud formation in woody plants, And so buds are induced to come out in the spring, and this is in combination with the temperature that also influences this process. So in fact, temperature and light are two complementary and overlapping abiotic effects that go together and regulate all of these processes, in fact. And I will make a little comment on that. And so here's a summary of the action spectrum. So here's the light source filtered through a prism in this case. And basically, there are multiple photoreceptors that are present in all plants that together integrate the different colors of light and are used to affect plant development in all of these processes I just told you about. And in fact, we have studied for many years the phytochromes. So in all plants have three phytochromes, A, B, and C, as photoreceptors that sense red light. Actually, they sense red and far red light. And then there's several plants that sense blue and near UV light. The cryptochromes, there's three of them in plants. Phototropins, which the name indicates are involved in phototropic behavior. Zeitlupe and FKF1 are regulators of flowering response. And so they're sensing the blue light in inputting into flowering. And then there's even plant photoreceptors that sense UV light. And what's common to all of these is that they have pigments that are present in the protein, which doesn't absorb light. So proteins don't absorb visible light, so they're colorless solutions, but they impart light harvesting by having a pigment associated with a protein. And of course, rhodopsin, you've heard about in light, and rhodopsin has a protein, which is called opsin, and a pigment, which is retinal. And those combine to allow this protein to sense visible light. So there really are two types of major pigments in plants that are involved in light sensing in protein photoreceptors. The first are flavins. So riboflavin you've heard about, right? And it's a B vitamin. And flavins actually absorb light in the blue region of the spectrum, and they're affected by their environment. And you can see here phototropism in seedlings, first described by Darwin, is due to the presence of a protein which binds a flavin, shown here, and that's called phototropin. And these are mutants here of this particular plant variety that lacks this particular photoreceptor gene, so the protein encoding gene for phototropin, and they do not respond to blue light, and that's how we know. Another photoreceptor that's blue light and uses a flavin is cryptochrome, and most plants have two cryptochromes, some have three cryptochromes actually. And you can see that if you lose cryptochrome 2, they behave pretty similar to wild type, but if you're missing cryptochrome 1, those plants can't sense the blue light and tend to grow as if they're etiolated and they're growing out much taller. Their hypocotyls are extended. So cryptochrome 1 regulates seedling establishment, whereas cryptochrome 2 has drastic effects on flowering, and this is just showing under long days wild type flowers, and without cryptochrome 2, the plants are delayed and they remain vegetative for much longer periods of time. This is just the absorption spectrum of the flavin, And what happens is the flavin absorbs light and reacts with a cysteine, a residue of the protein, and forms this linkage, which changes the spectrum. And the details of that aren't really important, except to say that it's a photochemical change in the pigment, which alters the protein, which then transduces the signal that leads to the change in growth or development in this case. So flavin-based photosensors are widespread, as I've told you, and all plants have this. And this is pretty much why you need blue light for robust plant growth and development. And this basically shows you that phototropins mediate a response, the low fluence response, where the chloroplasts will accumulate under low fluence of light. And under high fluence of light, you can see that you lack this second response where you can see high fluence. If you're missing phototropin 1, the chloroplasts move to hide underneath each other and protect each other from too much damage. Whereas in phototropin 2 mutants, there's really no effect of the accumulation of chloroplasts. And so this is how we know that these particular protein encoding genes are involved in these processes, is by identifying mutants and then studying their behavior in the absence of the particular gene. Now, let me get to the coup de grace, which is really my favorite photoreceptor, and that is phytochrome. And I had this horrible name, linear tetraperol, that Lucy was struggling on, but you did really well. It's really, if you look at the structure of this, it's not important to know, it actually is very similar. It's related to chlorophyll in many sense, but chlorophyll's a ring of 4 of these perols, and this is a broken ring, and it's we call them bilins. They're linear tetraperols. They're ring-opened porphyrins, for those in the know. And these were first actually discovered in humans as biopigments. And I think you've all heard of bilirubin, right? Bilirubin is a bilin. Although this is bilirubin's cousin, and these are found in plants. And these pigments are associated with proteins, in this case, the phytochromes. And what phytochrome is a photoreceptor, and I'll show you this spectrum on the right, that have 2 species. The PR, which absorbs red light, so I'm just putting the absorption right here, so they absorb highly in red, show the bright red above it. And when it absorbs light, this decreases and you get increased absorption of far red light. So it changes color upon light. And this, when it receives far red light, converts back to the PR state. And so what the plants do is that they measure the ratio of red to far red light. Because if you have more red, then you make more PFR. If you have more far red than red, as is the case in shade, you have more PR and the plant grows. So the plant is measuring the ratio of these two, the ratio of the wavelengths of light. And in fact, this ratio is perfectly set up. So and this is how it does it by just flipping this double bond. So very small change in this pigment, and it's very analogous to rhodopsin, retinal and rhodopsin. Same sort of process. And it's optimized, if you look, here's the action spectrum for CO2 fixation, and it lies right between these two. So it's tuned perfectly to find the ratio of light that's optimal for photosynthesis. And if it's off the optimum, then it changes the way the development of the plant to forage or to be happy and stay and remain vegetatively and short and robust if the light is appropriate. And you can even use this quantitatively. Here's the tomato series grown under different ratios of red and far red. And you can see that the stature of the plant, as well as the development and the petiole elongation and so forth, is depending on the red far red ratio. So going from simulated shade with a ratio of about 0.5 to just partial shade, 1.1. So plants are exquisitely tuned to measuring red far red ratio. And as you can see here, this is the spectrum up here. This is sunlight. So it has blue light through the visible and then out into the infrared past 700. And this green trace is shade. This is the transmission of light through a leaf. And you can see that the ratio of red to far red is about 1 for full sun, the yellow trace, and that ratio goes way below 1 in shade. And so plants have evolved this to sense the loss of light filtered through chlorophyll. So they're essentially the transmitted light through chlorophyll tells them, oh, there's other plants there. I need to do something. So the plants are seeing, and they grow and develop differently, as I've shown. Now, this is a little technical, but I wanted to say there's several phytochromes in plants. In fact, all plants have 3 phytochromes. And the reason they have 3 phytochromes is that they do different things. They can sense different qualities and quantities of light. The low fluence response phytochromes can sense red and far red. These are like the phytochromes that regulate seed germination. So red light, they make PFR, you get germination. Far red pushes it back, you get no germination. The VLF response are sensed by a different phytochrome that sense very low fluence of light. That's even below the amount of light you need to make chlorophyll. So these are very sensitive light responses, and pretty much any light will cause this response. Red, but they're maximally responsive in the red. And these are mediated by a different phytochrome. These are mediated by Phi A, and this is an HIR, high radiance response. This is continuous far red light, which cycles phytochrome through, and these responses have been shown through genetics to be mediated by this phytochrome A. So in fact, phytochrome A is important for shade tolerance. Its goal is to sense shade in this higher radiance response and sustain plant development when the main red far red sensitive phytochrome Phi B is causing the plants to just grow. So they sort of attenuate the strength of response to red light by phytochrome B. So essentially what I'm trying to say is plants have multiple genes because the phytochromes sense light in a different way. And I haven't shown Phi C here, but Phi C actually influences both, modulates Phi B activities and modulates Phi A, and plays a big role in the photoperiodic response of most plants. Now, I've studied phytochrome my whole life, and people say, wow, it's pretty important. But in fact, you can grow plants without phytochrome. And so we set up, a postdoc in my lab set aside genetic things to identify a pentuple mutant, Arabidopsis in this case, is a rosette plant in the mustard family. And you can see the most pronounced phenotypic change is seen in the Phi B mutant. But as you progressively knock out the different phytochromes, so this is a pentuple, this is a pentuple null, this is a quadruple and different quadruple, and here's a Phi A and Phi B. You can see that the growth and development of plants is dramatically altered when grown under red light, or white light in this case, with the presence or absence of phytochromes. But nonetheless, you can grow plants without phytochromes. They will make it, but in fact, they can't respond very well to diurnal light dark cycles. They need relatively high light to continue to grow. And these plants will actually grow and produce flowers and maybe make one silique and with a couple seeds, but they're not very happy. So plant growth and development is seriously impaired, and that's why we need phytochromes in plants. What was exciting in my lab is we did a mutant screen and we identified a mutation in phytochrome. It's just a point mutation. So the sequence, the protein itself was identical to the wild type, but it had one amino acid change out of 1,000 amino acids. And this particular mutation conferred light-independent photomorphogenesis. In other words, if you grew Arabidopsis in the dark, in this case, and many seeds, they won't develop. They'll just sort of elongate and run out of energy unless you're growing them on sucrose. But even with sucrose, they'll grow and they'll sort of eventually, they'll never develop true leaves. This particular mutation caused the plant to develop leaves, and this is grown in the dark, so you can see there's no chlorophyll in this. And these plants can survive in this unnatural condition with sucrose in the media for carbon because they can't fix CO2, and they will even produce flowers. So essentially, what plant growth and development is regulated is the process of starting from a seed, germinating, elongating, developing vegetatively, and then flowering is arrested at various stages of growth, and that there are inhibitors that prevent the plant from progressing through the cycle to flowering. And what this particular allele does is it allows it to bypass that light requirement, because this photoreceptor in these plants is able to function as if it's in the light and allow development to proceed all the way through the life cycle. So there's no arresting. Now, obviously, if you want a white crop that is achlorophyllous, if you're interested in cauliflower or something, or a different thing, this could be of interest. But again, who's going to try to grow plants with sugar? Okay, I'm going to skip this slide, but I want to emphasize that what phytochrome does is it reprograms the gene expression of the plant. So there's a wholesale change in gene expression that maybe 20 to 30% of the genome is altered by the sensing of light by phytochrome. So it's a huge regulation of gene expression, and that is arrested in the absence of light or activating of phytochrome. This says approximately 10% of the Arabidopsis genome was regulated light independently by this particular mutant allele that developed in the dark as if it was in the light. And here's some examples of what happens if you use this in different crop plant species. And you can see that this is a series of plants. This is tobacco, and this particular variety is shown expressing this particular mutant allele. And you can see that the plants are very dwarf and the flowering is delayed. And versus the wild type, which shows a much more elongated and growth habit. And these will be delayed flowering and are more dwarf. So this particular allele of phytochrome is a dominant allele, and actually we've generated it by classical mutagenesis in the endogenous position. So it's missing just one amino acid. It's altered in this case. You can see here that these plants shows severe dwarfing and severe sort of rosette growth habit, whereas wild type tobacco tends to elongate the inner nodes. So this is basically showing a lack of shade avoidance. They're severely non-shade avoiding. And again, you can see it here. This is Maryland mammoth. This is actually a short day plant. This plant won't develop flowers and was discovered because of its mutation until the days get very short. And I think they discovered this in Maryland. It was still growing without flowering when it started to snow. And you could only get it to flower by putting it in very long nights. Here is a plant. This is an isogenic line with this. So one can use this particular allele to regulate plant architecture. And you can also notice chlorophyll levels are quite different and that sort of thing. Phytochrome expressing this allele also affects leaf senescence, and so that's a relatively interesting. And it also affects vivipary. These, you can see seeds within the fruit of this particular microtom tomato is showing adventitious seed germination before the fruit is ripened. That's not really a good trait to have, but it's certainly interesting. Okay. So finally, I just want to talk about herbivory. Obviously, this is of great interest, and phytochrome actually plays a role in insectivory by regulating the program of plant defense genes that regulate or that provide tolerance to insectivory. And this is basically two plants with one of which has the YHB allele and the other is wild type. And this particular wild type plant is being eaten alive by aphids. And although it was growing in the same place as the YHB plant, this plant was severely stunted because of the aphid infestation. Aphids would jump around and you see them, but they just didn't want this plant. And so it's of great interest to potentially use this tool for regulating insectivory. And this is sort of the molecular basis. I won't really go into that. So that's about all I'm going to present today. Why plant C? And there's a lot there. So in addition to the photosynthetic apparatus, which is in the chloroplast, all plants possess a wide variety of flavin and bilin-based photoreceptors. These are the two pigment types that I just told you about, and allow them to sense the color, intensity, the direction, and the duration of exposure to regulate both growth and development of all plants. And obviously, these are really important components in the yield and agronomic properties of crop plants, as I've sort of alluded to. So these regulators are non-essential for survival. So plants can grow without these regulators, as I showed you, but they are really essential for successful competition in the natural environment, and also in the fluctuating environment, the diurnal environment, which of course um, is how they evolved to live on the planet Earth. And finally, these photoreceptors have overlapping and distinct signaling pathways, which regulate development in a wide variety of light environments. And people have been studying plants adapted to different kinds of environments and other competitions and competitors. And the phytochromes play, and the phytochromes and the flavin-based photoreceptors play an important part in the sensing of these. So basically we have a lot of knowledge of the genes involved and that they're varied in different cultivars of plants. So I'm going to leave you with this photo of Lake in Japan, where I took another sabbatical. And I just want to acknowledge I am retired as of 2020. Although I still have a program, I have some research support that is outlined down here. I'm supported still by the DOE and the NIH if they're still giving out grants in the near future. And but I have two senior researchers, Nathan Rockwell and Wei Hu, who are continuing to work with support from this on these projects. And I thank you for your attention, and I hope I've piqued your interest in how plants see and why plants see. So thank you.